Hey guys, it's Doc Janelle. Welcome back to my channel. The subject at hand is cytogenetics. And for this video, we will be talking about central dogma. It's very important for you guys to have a very solid understanding of this topic because not only because it will be included in your prelims exams, but because basically a lot of the other topics will be referring back to your knowledge on the central dogma. And also, you will be able to understand a lot of the diseases, the cytogenetic diseases, the cytogenetic problems later on based on their effect and the places where they cause damage in the central dogma. So anyway, if you're ready, then let's start. So what is the central dogma? The central dogma is actually a theory which was proposed by Watson and Crick in the 1950s. And this theory basically describes the relationship between nucleic acids and proteins as a directional flow of information. So what are our nucleic acids again? You have your DNA and RNA. And from our knowledge right now, we know that DNA produces RNA and RNA produces your protein, right? So that is the basic understanding that we have now. So basically, the central dogma describes that DNA, RNA, and protein has a directional flow of information. So pwede pala that protein can also produce your DNA, DNA can produce RNA, and then the circle just keeps going on and on. And pwede rin siyang reverse. That DNA can produce your protein, protein can produce your RNA, and RNA can produce DNA. So basically, it just talks about the detailed transfer of sequential information, no? From DNA to RNA to protein. Okay? So, huwag mo na kakabahan. Madali pa tong part na to. Alright, so the central dogma involves three important steps. These are transcription, translation, and DNA replication. So in this discussion, we will be talking about each of these important steps, what actually happens in each important step, and how they are related to one another. As we said, it is a sequence, no? A sequence, a flow of information. So in order for us to understand the central dogma, it is imperative for us to have a good understanding of the DNA structure. So a question, what are the building blocks of your DNA? Answer is your nucleotides. Yan. So nucleotides are the monomer units or the building blocks of nucleic acids. We have also talked about that your nucleotides are composed of three ingredients: your sugar, your nitrogenous base, and your phosphate group. As you can see in this picture, we have a phosphate group, we have a sugar, and of course, we have the nitrogenous base right there. Now, your nucleotides, the singular nucleotide, no? They have several functions already. So, even if hindi pa nabubuo yung DNA or RNA, meron ng function itong mga nucleotides natin. First, they function as coenzymes. So what are coenzymes? They are compounds which are required for catalytic activity. So they make sure that biochemical processes in the body actually go through. All right. So coenzymes help in the catalytic activity of the body. They also serve as donors of the phosphoric group. So, di ba ang isang example ng nucleotide natin is ATP and GTP. What does ATP stand for? Adenosine triphosphate. So, how many phosphate groups does this nucleotide have? So, from the name itself, triphosphate, so 3 phosphates. So, actually, this nucleotide is capable of donating one of their phosphates. So, if let's say ATP and GTP donates one of their phosphate groups, they will become, from ATP, it will become ADP or adenosine diphosphate. Di na lang because it has lost one of its phosphoric group. So, from 3, naging 2 na lang siya. Same with your GTP or guanine triphosphate. Okay? So, magiging GDP na lang siya. Now, aside from its capability of donating the phosphoric group, it also is capable of donating its sugars. Okay? So, yung sugar component ng nucleotide, capable din tayong i-donate yan. And also, it can also donate their lipids. Okay, ayan. Aside from this, your nucleotides also function as regulatory second messengers, which is important in hormone activation, and also in chemotherapy and immune suppression. So this structure here, this union of the phosphate, the sugar, and the nitrogenous base on its own already has several functions. Now, let's talk about the nitrogenous bases first. So the nitrogenous bases are nitrogen-containing heterocycles, okay? So they are ringed structures, itong mga nitrogenous bases natin. So they are cyclic or ring compounds containing carbon and obviously nitrogen, kaya nga nitrogenous base, and other elements. So they are, the nitrogenous base, guys, are the information containing parts of the DNA. So it is not the sugar that carries the information. It is not the phosphate that carries the information. It is the nitrogenous bases. So the sequence that the nitrogenous base create, yung pagkakasunod-sunod ng mga nitrogenous base natin, yan, that is what provides information. So if you remember, di ba, from our discussion of chromosomes, we mentioned there that certain parts of the chromosome has repeating sequences like A, T, T, G, C, C, P, mga ganun, di ba? So, yung pagkakasunod-sunod na yon, that is what carries information. Now, your DNA is usually measured by the number of base pairs. So, if there are 1,000 nitrogenous bases, we usually call it a kilobase. And if there are millions already of base pairs, we call it a megabase. Now, there are two types of nitrogenous bases, your pyrimidines and later on your purines. Yan. So, pyrimidines are 6-membered rings. So, let's count. 1, 2, 3, 4, 5, 6. So it's in a form of a hexagon containing 2 nitrogens. Where are the 2 nitrogens? You have 1 nitrogen here and another nitrogen over here. Now, your pyrimidines has 3 forms. Your cytosine, your uracil, and your thymine. So this should be thymine. I'm very sorry I wasn't able to edit this out anymore. So thymine. Now, in order to help you to memorize that, I put here an acronym, a mnemonic, which is CUT or CUT, so that you can easily remember your pyrimidine: cytosine, uracil, and thymine. Now, I also want you to remember that uracil is only found in RNA, while your thymine is only found in your DNA. Okay, so they usually exchange with one another. Kapag RNA, you will not have your thymine, instead you will have your uracil. Kapag DNA, wala nang uracil, instead you will have thymine. For your purines, it's actually composed of 2 rings which are joined together, forming a total of 9-membered rings. So let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9. And there are 2 forms of your purines, your adenine and guanine. Now, I've already mentioned this in our orientation or the very first topic, diba? That your base pairs actually pair up with one another. Sorry, your nitrogenous bases rather would pair up with one another. A pyrimidine would pair up with a purine. Okay? So the pairing ensures that there is a symmetrical double helix formation of the DNA. This tells us na yung sequences of nitrogenous bases on one side of the DNA has a complementary base on the other side. So let's look at this. Here we have adenine and its base pair is thymine. Always yan. Your adenine will always pair up with thymine and vice versa. While your guanine always pairs up with your cytosine and vice versa. Okay, so adenine is a purine while your thymine is a pyrimidine. Tama ba ako? Yan, tama. Syempre naman, tama, di ba? So, your base pairs are held together. They are connected to one another by your hydrogen bonds. Kindly remember that. I will ask that in your prelims. Okay? So as you can see here, meron ng arrow and label. The bond, the connection in the middle are made up of your hydrogen bonds. Okay? Now, let's go to your sugars. Your sugars are composed of pentoses. What do we mean by that? They are 5-membered rings of sugar. So let's count. 1, 2, 3, 4, 5. So usually what we count are the corners, no? Kasi yun yung nagsisignify nung ilang members yung meron yung ring. Okay? Now, what is the difference between these two sugars that make up your RNA and DNA? It is the attachment on the second carbon. So as you can see here, it's already in red. This is your second carbon. Okay? Now, your for the sugar for RNA is composed of ribose, and the attachment on the second carbon is a hydroxyl group, while the sugar in DNA is a 2-deoxyribose, which means that the attachment does not have oxygen, thus the name deoxyribose. Okay, so it only has hydrogen attached to it. Okay, so that is the one of the differences between DNA and RNA. Now, once you have your sugar and your nitrogen base together, it already forms a compound called nucleoside. Okay? So it is purine and pyrimidine attached to a sugar. Okay po? Now, what is that bond that connects the sugar to the nitrogenous base? It is a BN glycosidic bond. Okay? BN glycosidic bond. So, once a purine or a pyrimidine is attached to a sugar, it changes its name. Earlier, adenine, when it is attached to a sugar, you call it now an adenosine. A cytosine, together with a sugar, will be called cytidine. Your guanine, together with a sugar, will be called guanosine. And your uracil, together with a sugar, will be called a uridine. Okay? So, nag-change ng name. Later on, I will show you a slide showing the different nomenclatures or names kapag nucleotide, nucleoside, etc. Lastly, the last component of your DNA is a phosphate, which is symbolized by this blue ribbon that you see here on both sides of the DNA. So the phosphate forms the backbone of the DNA. So basically, this is where the sugar and the sugar and the nitrogenous base would attach to. So one of the phosphate chains would run from a 5 prime. So we call that apostrophe as prime. From the 5 prime going downwards towards a 3 prime direction. So one of those chains starts at the 5 prime and ends with a 3 prime, while the other chain starts with a 3 prime and goes to a 5 prime. So those are the two directions of your DNA chain. 5 prime to 3 prime and 3 prime to 5 prime. This is very important, especially when we start talking about DNA synthesis. Okay? And this opposing orientation of the two nucleotide chain is called anti-parallelism. Okay, anti-parallelism. What do we mean by this? Look at the starting nucleotides here. For the 5 prime, diba? The 5 prime is attached to an A T, a G, and a C, and then later a G and a T. Okay? Yan yung mga nucleotides attached to it. But when you look at your 3 prime to 5 prime, starting at the bottom now, it also has the same nucleotides. A, C, G, C, A, and T. Okay? So, parang binaliktad lang, di ba? yung components. Alright? So, we call it anti-parallelism. Now, what do we call that bond that connects your nucleotides to the phosphate chain? We call it an ester bond. Again, please do remember the bonds. I will ask that in your prelims. It's an ester bond. So here is the nomenclature that I was talking about. When you have your base pairs, we simply call them as adenine, guanine, cytosine, uracil, and thymine. But once you add a sugar to it, it changes its name to adenosine, guanosine, cytidine, uridine, and thymidine. If you add one phosphate to it, it will now be called adenosine monophosphate. If you have two phosphates, it will be called adenosine diphosphate. If you have three phosphates, it will be called adenosine triphosphate. Okay? So, yan yung pangalan nila. Or simply, shorten to AMP, GMP, CMP, UMP, and TMP. Alright? Now, putting everything together, your phosphate backbone, your pentose sugars, and their nitrogenous bases, this is now how a DNA would look like. Okay? So as mentioned, the genetic code is based on the sequence of your base pairs. Okay po? It is based on the order of your base pairs. And each strand, so this is one strand, di ba? One strand and then yung anti-parallel strand niya nasa kabila. Each strand has the same basic structure. Same lang sila ng structure. Phosphate backbone, attached to a sugar by an ester bond, and then attached to a nitrogenous base by a BN glycosidic bond. And then, paano nagdidikit yung base pairs natin? Hydrogen bond. So here is a diagram representation of the Watson and Crick model of a double helical structure. Okay, so ganyan yung itsura niya. Now let's go on to the first step of the central dogma, which is DNA synthesis, or also known as DNA replication. So this is a step which is needed for the production of new DNA, which is again, as mentioned in our last discussion, happens in S phase of interphase, di ba? Kasi kailangan natin ng new DNA for cell division. Now, it is important for you to remember that DNA synthesis proceeds in a 5' to 3' direction. So, kanina, di ba, we mentioned na yung isa dito, one of these strands goes from 5' to 3', while the other goes from 3' to 5'. Alright? So, as you can see here, the DNA replication occurs in opposite directions. So, let's say na itong strand from top is the 5'. So, 5' towards your 3'. And the other is 3' to 5. So, ang magiging replication niya will be opposite. Or, opposite. Kasi start siya sa 5', paakyat ng 3'. Yan, okay? So, there are requirements before DNA synthesis can occur. This is, first requirement, there should be a free 3' hydroxyl group. There should be a DNA template. And the DNA synthesis process is a semi-conservative process because each strand of the DNA will be kept. Diba? Like this one. Ito yung original DNA natin. Now, each single strand became a template for the new strand to form. So basically, we kept half of the original DNA. Kaya siya semi-conservative process. Now, here are the overview of the DNA replication. First, we have the parent DNA molecule. So, yun yung una. And then what happens is that the parental strand will unwind. When we say unwind, it will separate. Okay, it will separate and it will serve as a template for the new DNA that will be formed. So each parent strand will become a template for DNA polymerase to bind or to put complementary bases. So like this one, here we have a G that is exposed. So the polymerase should be able to find a complementary base for G. So ano ba yung complement base ng G? C. So ayan, kakabit yung C dyan. So kailangan complementary pa din sila. Also, the sugar phosphate backbones of the daughter strands eventually will close, and now you will have a new DNA. So let's basically dissect this overview of events. Step 1 is the unwinding of the DNA. So from being connected to one another, they will separate because of the helicase. The helicase is this pink structure right in the middle which somehow acts like a zipper. Ba siya yung nag-unzip doon sa DNA double helix natin. So it will first bind to the ori protein, which stands for the origin protein, and it will begin to separate the strands. Okay? So unwinding will begin at the AT-rich region, and what will keep it open are these pink proteins here on top and at the bottom, which we call binding proteins. All right? So once the helicase opens up the parent DNA, and magkaroon ka na ng dalawang single-strand DNA, what keeps it open are your binding proteins. Okay? Afterwards, you see this purple structure here. This is a primase. So the primase will start to create a short stretch of RNA on the DNA template. So as you can see, di ba? Sabi natin kanina, DNA synthesis occurs in a 5' to 3' direction. So first, the original strand on top is 5' to 3'. So ang DNA synthesis will occur from 5' to 3'. So basically, magsisimula siya dito sa pinakauna. Kasi ang complement ng 3' ay 5', di ba? So from 5', it will now create a new strand which is 5' to 3'. Para opposite siya nito. Okay? Hopefully naintindihan nyo yun. And then, yung primase sa kabila will create, kasi the template strand is 3' to 5'. So, kailangan in this direction papunta para opposite ng strand ulit. Kasi dapat 5' to 3'. So, habang ino-open, doon yung dulo naman ang nilalagyan. Okay? Ganun siya. Now, after we open up the parent DNA and we start to create a new RNA strand, ma-form yung replication fork formation. What is the replication fork? It is the appearance of the unwound DNA when it starts to form the leading and the lagging strand. So, yun daw. Ano ba yung leading and lagging strand? So, the leading strand is the strand on top. Okay? The original strand, the 5' to 3' strand. Siya yung leading strand. And the lagging strand is the strand at the bottom, or the 3' to 5' strand. Siya yung lagging strand. So we have a leading and a lagging strand. Okay? So as you can see here, medyo nagmumukha na siyang fork talaga, di ba? Tapos meron pa yung gitna. Yan, so mukha na siyang fork talaga. Afterwards, we have DNA polymerase, itong bean-shaped structure na ito. The DNA polymerase will be the one to add DNA nucleotides to the RNA primer. So as you can see here, the RNA primer are the ones in green. Okay? Green. Yan. Pero yung DNA nucleotides, they are the ones in blue. So, si primase yung nag-add ng RNA, pero si polymerase yung mag-add ng DNA nucleotides natin. Okay? Again, the overall direction of the replication is from 5' to 3' in the leading strand and 5' to 3' again in the lagging strand. So, one ability of the DNA polymerase, aside from adding your DNA nucleotides, is also to proofread yung mga base pairs na dinadagdag niya. So, kapag nakita niyang mali yung inad niyang base pair, it will replace it with the correct base. Okay? So, here is another picture showing you the DNA polymerase. So, it's actually a primary replicase enzyme. Again, ang role talaga niya is to perform the elongation of the DNA strand. So, what it does is it adds nucleotide una to the RNA primer. Diba? As mentioned kanina. And then, sunod-sunod na nilalagyan ng DNA nucleotides by the phosphodiester bond. Okay? Tapos yung sinabi kong proofreading ability niya, ang direction ng proofreading ability is from 3' to 5' naman. And ginagawa na yan through a sub-enzyme called exonuclease. So si exonuclease, yung component ng DNA polymerase that functions to remove incorrectly incorporated nucleotides. Okay? Yan. Now, as you can see, again, we have the leading strand and the lagging strand at the bottom. There is continuous strand synthesis, no, doon sa ating leading strand. Pansinin nyo, yung primer, RNA primer, tapos magkakadikit yung mga DNA nucleotides natin, di ba? Doon sa leading strand. Pero sa lagging strand, if you notice, RNA primer, DNA nucleotides, tapos may space. Tapos another RNA primer, DNA nucleotides, tapos may space. So, hindi siya continuous. And those fragments are what we call Okazaki fragments. Again, they are found on the template of the lagging strand. Okay? Later on, I will show you how magdidikit yung mga yan. So here is a good picture showing that, no? So the enzymes will then remove the RNA primers. Yung mga green kanina, if you notice, they are gone. Okay, so some certain enzymes will remove the RNA primers. And there is another enzyme called ligase. Ligase will seal the sugar phosphate backbone and will be responsible in joining the Okazaki fragments together. So si ligase, itong parang rectangular enzyme na ito. Siya yung pupunta doon sa fragment ng Okazaki fragment at pagdidikitin niya yan. Okay? So replication is completed by filling in the gaps by the DNA polymerase and your ligase. So afterwards, after we create two DNA molecules, we will be left with two extremely long DNA strands. So as we know, these DNA strands are very, very long. So we have to be able to condense them into a smaller structure in order to facilitate faster cell division. So yung protein that they need to attach to para maging condensed sila is what we call a histone. Okay? So, from being extremely long, they will condense and will become a chromosome which is only 2 micrometers long. Okay? And it shrinks by 7,000 times. No? Yeah. So, your histones, they resemble beads on a string. Kasi dyan iikot yung DNA strand natin. So again, just to review the different enzymes that were used in DNA replication. First, you have your helicase, which acts like a zipper, opening up your parent double helix DNA. You have the binding proteins, which keeps the strands separate and open. The primase, which adds your RNA primer or template strand. Your DNA polymerase, which has two roles, diba? To add your DNA nucleotides, and yung proofreading niya through exonuclease. And then lastly, your ligase, which joins together your Okazaki fragments. So that is how DNA replication occurs during the S phase of your interphase. Next, we have RNA. So RNA is actually a polymer composed of purine and pyrimidines attached to a ribose sugar, di ba? And they are linked together by a 3' to 5' phosphodiester bridge or bond. Okay? So, here are the differences between DNA and RNA. Be able to memorize this. I think there are 2 or 3 questions about this in your prelims. So, DNA is a double-stranded structure, while RNA is usually single-stranded. DNA has thymine, and in RNA, thymine is replaced by uracil. The sugar of DNA is deoxyribose. It does not have oxygen in the second carbon, while RNA has ribose as a sugar. Your DNA contains protein encoding information, while your RNA also has protein encoding information, and it controls how the information will be used. DNA cannot function as an enzyme, while RNA may function as an enzyme. Your DNA will persist while your RNA is short-lived. Here are the different RNAs found in our cells. First, you have your rRNA, or also known as the ribosomal RNA. Pero basically, tinatawag lang natin yan sa cell na ribosome. Okay? Ribosome lang siya. Remember, yung nakadikit sa rough endoplasmic reticulum, your ribosomes. Your mRNA or messenger RNA, yan yung product ng transcription. Okay? Siya yung may dala-dala ng protein information. Yung tRNA, also known as transfer RNA. Why is it called transfer RNA? Because it transfers the amino acid later on, malalaman natin. And then last year, small nuclear RNA, which is not important in this discussion. So, mRNA or messenger RNA, as mentioned, it is a chain of nucleotides. Diba? Again, it is the end product of transcription. When we say transcription, a copy. So, we made a copy of a portion of the DNA, and that copy is the mRNA. So, it copies the DNA and then leaves the nucleus through the nuclear pore. And being RNA, the nitrogenous bases are only A, G, C, and U, and there is no thymine. So here is an image of your mRNA. So your mRNA contains information in order to create a specific protein. Okay, so yung bases ng mRNA natin, it codes for several amino acids. And when you put together your amino acids, you will create protein. So your mRNA is composed of 500 to 1,000 nucleotides, and it is read by a sequence of 3 bases. So every 3 AGCU, we call it a codon. Okay, a codon. And usually, the very first codon is AUG, which codes for methionine. Methionine is an amino acid. Please do remember that AUG, which codes for methionine, is also your start codon. And then, yung end ng mRNA natin will either be a UAA, UAG, or UGA. Or simply, uwa, wag, uga, which are your stop codons. So, yan yung last codon that you will see in an mRNA. Second, we have the ribosomal RNA, also known as your ribosomes. Okay, so an rRNA is also a single strand composed of 100 to 300 nucleotides long, but it is globular in its shape. It may be found inside the nucleus, or it is made rather inside the nucleus, specifically in the nucleolus. Kindly remember, it might just be asked in your exam that the nucleolus is not always found in the cell cycle. Okay, usually nag-a-appear lang yan after cell division. So, the ribosome will associate with proteins, okay? And it helps in protein synthesis. We have your tRNA also, or transfer RNA, which is clover leaf shaped. And it is a single-stranded molecule with an attachment at the end for an amino acid. As you can see here, no? Yung dulo niya is attached to an amino acid. And then the opposite end, so yung opposite end ng amino acid also has three important bases, and we call it the anticodon. So ito, yung three base na yan at the end of the tRNA will attach, or should be complementary to the codon of your mRNA. So later I will explain that. So again, your mRNA, ribosomal RNA, and transfer RNA. So here is how they will all work together. We have your mRNA, or the copy of your DNA. It will be read by your ribosome. And as you can see, we have the tRNA. One end is attached to an amino acid. The other end has the anticodon. Tapos yung anticodon is complementary to the codon of your mRNA. So here, AGU. So ano ba yung complement ng A? U. And then complement ng G, IC. Complement ng UIA, so UCA. So mag-attach yan dyan. Pag nag-attach siya, the amino acid will go to the growing protein. So now let's go to transcription. Transcription is the first step in gene replication. Remember that. I think I asked that in the exam. First step in gene replication. So because the DNA is too big to exit the nucleus and form a protein, a copy of the DNA should be made, and it will be the one to exit the nucleus and go to the cytoplasm. Basically, that is the process of transcription, making a copy of the DNA so that it can go to the cytoplasm and create the needed protein. So, the DNA strand will split, and what will open that DNA in transcription? The RNA polymerase. Kindly remember, sa DNA synthesis, ang nag-open ng DNA is your helicase. Pero in transcription, what opens your DNA is RNA polymerase enzyme. As you can see here, siya yung nag-open or nag-unwind ng ating DNA strand. After ma-open, complementary nucleotides will now form on the template strand. So as you can see here, the bottom strand became the template. So TAC. Tapos if you look at on top, yung red, yan yung mRNA, may complementary base pair. Yung T, complement niya is A, complement ng A in RNA is U, complement ng CIG, etc. So on and so on. So, your transcription occurs in 3 stages. Initiation, again started by your RNA polymerase, and then elongation of your mRNA, and then termination of that mRNA. So, initiation is the control point that determines which genes are transcribed. Okay? Initiation determines anong part ng DNA yung ikakopy. And then RNA nucleotides are added during elongation. And then lastly, terminator will signal the end of transcription. So now, after transcription, we now have an mRNA which will exit the nucleus through the nuclear pore and will go to the cytoplasm. Okay? Pagdating niya ng cytoplasm, pupunta na siya ngayon sa rough endoplasmic reticulum where it will do translation. So in translation, yung mRNA, which is the message, diba, the copy, it will now be translated or interpreted to form a protein. So each amino acid is formed by 3 nitrogenous bases. Pakita ko lang. Yan. So each amino acid, so here we see the amino acids phenylalanine. So ano yung 3 base pairs niya? UUU and UUC. Yan. So each amino acid is composed of different triads or codons of your nucleotide. So let me just go back to the slide where I was earlier. Yan. So it's composed of 3 nitrogenous bases. So in translation, the ribosome will read the mRNA in groups of 3, and the anticodon or the complementary codon in your tRNA will match on the RNA or the mRNA to give a specific amino acid. Kung nalilito na kayo, i-YouTube nyo to, okay? So basically, ayan, we have the mRNA at the bottom. This brown structure is your ribosome. So si ribosome, mag-a-attach siya dyan, as you can see here, at babasahin niya yung mRNA natin. So nabasa niya, AUG. So siya yung magsasabi kay tRNA, kailangan ko ng complement kay AUG. So si tRNA, dadalhin niya yung complement ni AUG. What is the complementary of AUG? UAC. And again, ano nga yung AUG? It is our start codon. And what amino acid does it begin with always? Methionine. So, hahaba na nga haba yung amino acid na yan until maging protein siya. So, lastly, last slide na to, protein synthesis, again, is formed through transcription and translation. Your amino acid is formed by 3 nitrogenous bases. And once the RNA reads the stop codon, that's the end already of your translation. And voila, you have your new protein. So if you got a little confused about this, I suggest you watch some YouTube videos to basically summarize everything that you have just learned in this discussion. Study hard. This is 30 points on your prelims. Good luck and God bless. Bye!